good evening, everybody. My name is Tim Pedley, and I'm a Emeritus Professor in the Department of Applied Mathematics and Theoretical Physics here in Cambridge. My research has, for the last, must be nearly 60 years, been in the application of fluid mechanics to biological and medical problems. In both internal flows, such as blood flow, which I'll be talking about later on, and external flows, such as those generated by swimming organisms, which is actually what I mostly work on these days. Anyway, I hope you will enjoy it.
Welcome to the third lecture in the 2021 Darwin College lecture series on blood. And I must say we are particularly pleased at how many of you have joined us online for the previous two lectures and indeed many others who have looked at them afterwards. In our first two Darwin College lectures this year, we've heard from a consultant hematologist, Claire Rohde, about the history of treatments for leukemia and especially her own work on a novel new therapy, and from a literary historian specializing in women's reproductive health, Sarah Reed from the University of Loughborough, who provided us with a fascinating insight into the evolution of the cultural and literary representations of the reproductive female body in early modern England. This week, we are really fortunate to be able to add a perspective from an applied mathematician to our understanding of blood. Our speaker today is Professor Tim Pedley, the G.I. Taylor Emeritus Professor of Fluid Mechanics in the Department of Applied Maths and Theoretical Physics here at the University of Cambridge. Tim has pioneered the application of fluid mechanics to understanding biological phenomenon. His best-known work includes the study of blood flow in the arteries, flow structure, interactions in elastic tubes, and flow and pressure drop in the lungs, as well as the collective behavior of swimming microorganisms. As I've said before, we are of course sad not to have a live audience here in Lady Mitchell Hall in Cambridge, but we are really grateful to so many of you for joining us online. And we thank our speakers for making the time to pre-record their lectures in lockdown with such challenging tasks that brings for many. The lectures, of course, will remain online, so please do share the details of our YouTube channel with anyone you think might enjoy today's talk. The title of Tim's lecture is Blood in Motion, the Physics of Blood Flow. I hope you enjoy it. Thank you.
Well, good evening again, everybody. This talk is, as you can see, on blood in motion, the physics of blood flow. It's a great honor to be able to give this Darwin lecture, and thank you all, the college, for inviting me. Now, we all know these days that the blood circulates. This is a sketch of the circulatory system. It goes in two loops. Most of the body is served by the systemic circulation, which is powered by the left ventricle, which I'm pointing at, and then that ejects the blood into the aorta, the main artery, and that goes on through a sequence of increasingly branching vessels, smaller arteries, whose net cross-section is increasing all the time, until you get down to very small arteries, and then the capillaries in the microcirculation, and it's across the walls of the capillaries that mass transfer takes place. The cells in the tissues and organs are provided with oxygen and nutrients, and they get rid, and they do things with them, making energy, proteins, whatever, and they unload the metabolites that they produce, especially carbon dioxide, are put back, and the Then it goes into the venous system, and small veins go to bigger veins, etc., etc. Eventually in the vena cava, which delivers the blood to the right atrium of the heart. That puts it in the right ventricle, which when it contracts, drives the flow into the pulmonary artery, which goes through the lungs, a parallel system of microcirculation, where the CO2 is unloaded and the oxygen supply is topped up, and then it comes back, reoxygenated blood comes back into the left atrium, which then supplies the left ventricle, etc. It takes a particular blood cell about a minute to go around the whole system on average. You'll note that there are valves at the inlet and exit of both the ventricles, non-return valves, so that when the ventricle contracts or doesn't contract, fluid backflow is forbidden. But people didn't always know this. It was William Harvey in the early 17th century who discovered and promulgated his discovery of the circulation of the blood to people in England and Europe. The medical education that Harvey and his contemporaries had received was all based on the ancient Greek system, of who the most famous member of which is Galen in the 2nd century AD, who knew a lot. He understood the functions of the 4 heart valves, the variation, he knew about the variation of the pulse, he knew the different structures of the walls of arteries and veins, but he didn't really know what the blood was for. It was thought that it sort of flowed backwards and forwards in both arteries and veins, being regenerated, perhaps in the liver, every heartbeat. The driving force was supposed to be the squeezing and relaxing of the central vessels as a result of the contraction and expansion of the thorax during breathing. But he didn't know about the circulation. Actually, unknown to Galen, ancient and traditional Chinese medicine talked about circulation of the blood in the 3rd century BC. The Huangdi Neijing said that the blood current flows continuously in a circle and never stops. It may be compared to a circle without beginning or end. And also in the 3rd century, there was the famous publication, The Pulse Classic by Wang Shuhe, who explained, and lots of people learn from it, that the pulse is examined not just at one place on the wrist, but at 3 and with varying degrees of pressure. Here's a picture of an ancient picture and a modern picture, because traditional Chinese medicine clinics still exist. And here's somebody with 3 fingers applying different degrees of pressure on 3 different bits of the vein in the arm. And it is claimed that they can diagnose problems in the heart, the liver, the kidney, other organs, and even, it was said, predict the time of death, although I'm not actually aware of a successful test of this system using modern quantitative techniques. And now another person who came before Harvey and went a long way to discovering the circulation was Ibn Ben Nafis in the 13th century, who was very active in the golden age of Arab science. And he was the first person, for example, to describe the pulmonary circulation through the lungs. And for the blood to be able to circulate, it was necessary that he would propose the presence of small communication vessels between the arterial side and the venous side, which predated by about 400 years the first observation of the pulmonary capillaries by Malpighi. But anyway, his work, like that of the ancient Chinese, was not known in the West. And it was Harvey who discovered it. I should mention that in the West, there were people who did a lot of work on anatomy and physiology of the circulation. And these pictures of the heart by Leonardo are remarkable. They show a heart with the four major vessels that go in or come out. It shows a valve in the aortic valve. It shows arteries on the surface of the heart, which we know to be those that supply nutrients and oxygen to the heart muscle. But it's very similar to modern day sketches of the heart and the ventricles and atria. But Harvey proposed that the blood circulates, and he proposed it in a book, Exercitatio Anatomica de Motu Cordis et Sanguinis in Animalibus, an anatomical treatise on the motion of the heart and blood in animals, published in 1628. And probably the most persuasive arguments that he used for others, it wasn't all that he wrote or did, but the two factors that made it inevitable that the blood had to circulate, I think, were the following. First of all, there are non-return valves in the veins of the limbs anyway, and you can show this. You put a ligament round the upper arm, not tight enough to stop venous return coming through it, but not so tight as to stop the arterial blood going into the arm. And these, the veins on the arm bulge up, and these little blobs are where the valves are. And if you stick his finger on a valve, he showed that the blood in that vein disappears from the region between that and the next blob. The next blob being a valve. And even when the ligament is, ligature, is released, and he keeps his finger on the valve, the blood in these veins near, more central to the finger, goes away, but doesn't come back into the bit of the vein that it had gone away from before, until he releases his finger, and then it does. So non-return valves means that the blood flow must be in one direction only in the veins, and he inferred in everything else. He also made an estimation of what we would these days call cardiac output. He said that the heart had to pump about 36 ounces, which is about a litre per minute, which is less than the modern figure of 5 litres a minute, but far too much to be created each beat in the liver, by what Harvey said as by the juice of ingested food. Now, my topic is how the blood is pumped round and flows round in the body. The force that generates flow has to be a difference in pressure between two ends of a tube, any tube. And the first person to measure blood pressure was Stephen Hales in the early 18th century. This rather drastic experiment. He measured arterial blood pressure in many different mammals, and the horse is the most famous one. This was a horse which was going to be put down anyway, but it still seems pretty drastic. Anyway, he made an incision in the carotid artery and connected it to a long glass tube which was held vertically, and then he looked at how high the blood went up that tube, and it went in the maximum blood pressure. It pulsed, of course. Things pulse in arteries, as we know. It pulsed, but the maximum that it went up to was greater than 8 feet, which is equivalent to, in modern terminology, 180 something millimeters of mercury, which is a somewhat high, but not outrageously high systolic blood pressure in mammals generally, including people. Whereas in the veins, it was easy to show by cutting into them the blood pressure was much lower because it sort of dribbled out. And people were familiar with some aspects of this because of bloodletting, or breathing a vein, as it's called. It was a popular treatment for feeling unwell, both because of, as the medic said, it would make you feel better on theoretical grounds based in Galen's theories of the humours, which I'm not going into, and presumably it was also popular because when you lose a little bit of blood, for instance, when giving blood these days, there's a temporary euphoria that it can generate. This fellow doesn't look very euphoric because it looks to me as if he's been stabbed in an artery rather than the vein, which is why it's squirting out quite vigorously and not dribbling out as it would in a vein. Now, okay, the blood, we've got blood pressure, high in the arteries, low in veins, and it pulses. Now, Euler, the famous Leonhard Euler, who started so many areas of mechanics, fluid mechanics, solid mechanics, and many other things as well, realized that the pulse must propagate along the elastic arteries. And I'm going to talk about the mechanics of that propagation in a little while. And he correctly wrote down the governing equations. There are really just two. One is concerned with conservation of mass, and the other is based on Newton's second law of motion. But he failed to solve them, much to his chagrin, because of unrealistic assumptions about the properties of the vessel walls. That may be why he's looking grumpy in this picture. And it fell to Thomas Young, the English polymath, to correctly predict the speed of propagation of the pressure pulse in arteries. In his Croonian lecture in 1808 to the Royal Society on the functions of the heart and arteries. And he was a marvelous person who did all sorts of things, not just talking about heart and arteries. His derivation of the wave speed is correct, but not easy to follow because he relies on rather obscure physical arguments rather than solving the equations that Euler had written down, and as we do these days. But I can't resist one quotation from his Croonian lecture. The inquiry in what manner and in what degree the circulation of the blood depends on the muscular and elastic properties of the heart and of the arteries, supposing the nature of those powers to be known, must become simply a question belonging to the most refined departments of the theory of hydraulics. And as an emeritus professor of fluid mechanics in the Department of Applied Mathematics and Theoretical Physics, I reckon that's one of the most refined departments of the theory of hydraulics as well. Right, but the modeling or the theories of Young and Euler did not include the frictional or viscous properties of blood. They obviously knew about it because there's a big, squeezing blood through lots of little capillaries, which they all knew about by then, would obviously create a lot of resistance. But it wasn't important, they said, in the propagation of the pressure pulse in arteries. So let's go down to looking at resistive properties of flow in tubes. And here we come to Poissons, who in Paris in the middle 19th century did some very exhaustive and careful experiments on water flow in very narrow tubes. And he found experimentally that if the tube has diameter d, length L, pressure drop from one end to the other, P1 minus P2, then the flow rate, the volume flow rate, is proportional to P1 minus P2 and to the fourth power of the diameter. So that when the diameter goes down, the flow rate goes down a lot, and inversely proportional to the length. And the constant K is a constant that depends on the fluid used and the temperature, but not on the tube. The theory for that was done properly by Hagenbach in 1860, based on the recently codified Navier-Stokes equations for fluid flow, which introduced the viscosity of the fluid, and his theory predicted the constant K, and so we get what is now the well-known Poissons Law, with mu, the viscosity of the blood, being in the denominator. But let me emphasize what that theory was. It was for steady flow in a long, straight, rigid tube far from the entrance. Every fluid element, every piece of the blood moves in straight lines with no acceleration, which means there's no net force on it. The pressure gradient force driving it through is balanced by viscous stresses. The velocity profile across the tube becomes parabolic, with the central velocity being twice the mean velocity. And the important thing about why there is a profile is the fact that all fluids adhere to, through molecular forces, to any solid that they are flowing over. It's called the no slip condition. So the velocity had to be zero at the walls and larger in the middle. So that was a satisfactory theory. It agreed with the experiments. And I should have said that Poissons himself was a medic and was interested in the resistance to blood flow in small vessels. But he rapidly discovered that blood has an unfortunate tendency to clot when you try and do experiments with it. So he, fortunately for us in mechanics, did his experiments with water. And in any case, Poissons flow does not occur in any blood vessel. Why not? Well, because this is a sketch of the systemic arteries in a mammal, probably not human, since it appears to have a tail. But like humans and any other mammal, the tubes are not long. They might be long, but they're not. There's no great length between one branch and the next. They're not straight, especially the aorta as it comes out of the heart. Curved, has a big curve in it. The vessels are not rigid. The flow is not steady because of the beating of the heart, and some vessels, the overall geometry is unsteady. These are arteries on the surface of the heart, and they will pulse in and out, change their curvature as the volume of the heart changes during the heartbeat. And moreover, blood is not a continuous liquid. It is a suspension of blood cells, mostly red blood cells, in plasma. The red cells are flexible biconcave discs. You see a lot of them pictured here, approximately 8 microns across, and they occupy about 45% of the fluid volume. And this is such a high volume fraction that the blood wouldn't flow at all if the cells were not highly deformable. However, in blood vessels whose diameter is much larger than that of the cells, whole blood behaves as if it is a normal continuous fluid called a Newtonian fluid in the jargon. But when you're down in the microcirculation, in the capillaries, red blood cells have to be deformed just to fit in, and they go down in single file. This is a narrow capillary tube of about 6 microns in diameter. So you have to squish the cells to go down that. And in between, there are, of course, a whole lot of vessels that eventually branch into teeny capillaries, but before them, they're in what are called arterioles, which tend to be, which are like very thick-walled, very small arteries. They have a lot of muscle in the smooth muscle in the walls, which when it contracts can control local blood flow from one bit of the circulation to another. This picture was taken of the microcirculation in the mesentery of an animal, which is a thin sheet of tissue which is attached to the intestines to help stop them flopping about, basically. But that means it's very easy to see through and get pictures of. So in the dynamics of blood flow, we've said that it's not, well, we've said that Young and Euler could ignore the viscous properties in the arteries, but we've seen that it has to be important in the microcirculation. Let's consider that a bit more physically by talking about inertia. Inertia is the property of matter to go on moving with the same speed in the same direction unless acted on by a net force. This is Newton's first law of motion. And then it experiences an acceleration and gains inertia, Newton's second law, which depends on its mass. The second law is often summarized by the equation F equals ma, force equals mass times acceleration. Now, in the case of liquids, rather than gases, so liquids are almost incompressible fluids, so we can forget about the fact that gases are compressible. For the case of liquids in tubes, the force consists of pressure gradients that drive the flow and may accelerate it, and viscous forces that resist the fluid's shear and dissipate energy. There may also be external forces such as gravity. The relative importance of inertia and viscosity is often estimated by calculating something called the Reynolds number, which is the ratio of a typical mass times acceleration, or inertial force as it's called, to a typical viscous force. And for flow in a tube, the Reynolds number is defined as the density of the fluid rho, the diameter of the tube, the average velocity in it, on the numerator, and in the denominator, the viscosity. And that's a dimensionless number. If you have consistent units for all the items, this thing doesn't have units. It's not a length or a time or a force or something. It's dimensionless. And that's a very useful. And just to show you, you're not supposed to memorize everything in this table. There's no test at the end. Showing the typical dimensions, velocities, Reynolds numbers in blood vessels of a dog, actually. Humans have rather higher, larger aorta, for example, than dogs, etc. And the Reynolds number is calculated assuming a viscosity of 4 millipascal seconds, which is 3.8 times the viscosity of water. Anyway, you see that the large arteries, aorta, different bits of the aorta, the femoral artery, have a diameter of about 15 millimeters, so 1.5 centimeters. In humans, it's about 2.5. Getting smaller as you go past through lots of bifurcations and branches, and you've got smaller dimensions. But they're still that order of magnitude. And the typical peak velocity in the large arteries is of the order of 1 meter per second. And therefore, the Reynolds number in the large arteries is measured in the thousands, which is large. In the microcirculation, the arterioles and the capillaries, the Reynolds, they're much smaller with lower velocity, and the Reynolds number is much less than 1. So in arteries, the Reynolds number is large, and inertia is much more important than viscosity. Whereas in the microcirculation, the Reynolds number is small, and inertia is unimportant. But don't forget that anyway, you don't get Poissons flow in the microcirculation because of the large volume fraction of suspended cells. It is found that this is now a sketch of time, how a very qualitative sketch of the variation of pressure with time in different parts of the circulation. So in the ventricle, the left ventricle, the pressure, by the way, the pressure is always measured relative to the exterior pressure at the level of the heart. So in the heart, the pressure goes from 0 in diastole to 120 millimeters of mercury in systole. When it goes through the aortic valve into the aorta, it injects fluid in there, and you get a pressure pulse, and we're going to talk about the propagation of that, which changes shape somewhat as you go on down. But in all the large arteries, the mean pressure, and indeed the amplitude, the height of this wave, stays more or less constant. It's only when you get to very small vessels, especially the microcirculation, both the mean pressure goes right down and the amplitude goes right down as well. So in the microcirculation, there's hardly any pulsatility of the pressure. And that, of course, it results from the viscous properties of the blood. Now let's look at the propagation of the pressure pulse a bit more closely. These are measured curves of blood pressure in millimeters of mercury against time in the aorta of a dog, I think, measured with a catheter, which is pushed up inside the aorta. And and I should say that each of these curves are triggered. The time t equals 0 is a particular time in the electrocardiogram. When you're very near the heart, as soon as the valve opens, you get a steep rise in the pressure in the aorta, and then it goes on rising and then eventually decays. If you go a bit further downstream, the same thing happens, but later, 4 centimeters downstream, it doesn't start rising until a bit later. And then each of these curves is of 4 centimeter differences. There's a 6 there that should be an 8. So in other words, the wave propagates. These curves get a bit closer together as you go further downstream, which means that the wave speed increases. So you get from one place to another place 4 centimeters away more quickly. But I suppose the overall dominant feature of this, apart from the propagation of the wave, is the fact that its amplitude increases as you go away from the heart in the aorta. And it also gets a bit steeper at the front here, but that's a small effect. So when the heart ejects the contents of the left ventricle into the aorta and distends it, and here's some more sketches of not only the pressure pulse in the aorta and the femoral artery, but also the flow velocity waveform, which is different in shape from that and decreases in amplitude. So, as I was saying, pulse wave propagation begins with the expansion of the elastic aorta. And like any other oscillatory phenomenon or wave propagation, it comes about through a balance between the restoring force, which in this case is the elasticity of the vessel, which tends to contract again when it can after being expanded, and blood inertia. In the case of water waves, it's gravity and water inertia. But any oscillation of a pendulum which swings at the extremity of its oscillation, there's no speed, but the weight experiences gravity and therefore is restored, comes back, but it's going quite quickly when it gets to the bottom and overshoots because of inertia. Now, the theory, Young and co, based on conservation of mass and Newton's second law, leads to a prediction of the wave propagation speed. I won't go through the calculation, although if you were an audience of mathematics undergraduates, I certainly would. It's a very simple one. If the artery has diameter d, wall thickness h, small thickness compared with d, the Young's modulus of elasticity E, that's the same Young, he did work on elasticity as well, and the blood density rho, then the simple theory gives that the square of the wave speed is E times h divided by rho times d. This formula was essentially what Young got, but didn't do it very mathematically, and it was published by Mearns and Korteweg in the 1860s later on the continent. And the good thing about this theory, it's very simple. but it gives predictions that agree with experiment. If you independently measure E and H and D for the relevant blood vessels, you come up with, you predict a wave speed of about 5 meters a second in the proximal aorta, and that's about what the wave speed is in the proximal aorta of most mammals. As you go more peripheral, the speed increases, as I showed in the aorta, to about 8 meters a second, and this also agrees with measurement. So that's ideal applied mathematics, really. You've got a theory, very idealized. It ignores all sorts of stuff, but it predicts the thing you wanted to predict, the wave speed, pretty accurately. By the way, before I leave this slide, just to note that the wavelength of a wave is the wave speed divided by the frequency, and the frequency of, in dogs and people, is about 1 hertz, 60 beats a minute. So the wavelength is several meters, much longer than the length of the system, but it's still important that the wave is propagating. But anyway, it was a good theory. It predicted the wave speed, but it also predicts that there is no change of the shape of the pressure wave as it propagates, and that there's no difference between the shape of the velocity and the pressure waves. And neither of those is true. Most of the discrepancies can be accounted for by considering the fact that the arteries are branched and you get reflections of waves whenever they come across an obstruction. And the things that do come out of that theory are the peaking of the pressure pulse and the shape of the velocity waveform. The steepening at the front is a nonlinear effect, which I shan't be talking about it, and the effect of viscosity is present but not dominant. So let's look at wave reflection for a moment. So let's take a simple model of a bifurcating tube. You can think of this as the abdominal aorta and the femoral arteries, but it's straight and semi-infinite, etc. So we'll suppose that there's a pressure wave incident on this bifurcation. When it gets there, some of it is reflected and other parts of the energy are transmitted. Let us take the amplitude of the incident pressure wave to be p sub I, and what we want to predict is the amplitude of the reflected wave, p sub r. We should note, and this is something else you're going to have to take on trust, is that if the pressure wave amplitude is p sub I, the flow rate amplitude for the incident wave is y sub 1 for vessel 1, times p sub I, and y is the ratio of the cross-sectional area with rho the density and c the wave speed, and that's called the admittance of the vessel. It's the inverse of the impedance, if you like that better. And there are two conditions you have to apply to represent the junction. One is there must be conservation of mass. So any net flow into the daughter tubes must be the same flow rate going out. And the other thing is that the pressure must be continuous, because if it wasn't, there would be a very high pressure gradient over a very short element of liquid, which would therefore, by F equals ma, experience a very high acceleration, which is not observed. And when you do all that, you get a prediction of the reflected amplitude to the incident amplitude given by this formula. The total amplitude at the bifurcation in tube 1 will be the sum of the amplitude of the incident and the reflected wave, which will be greater than the amplitude of the incident wave if the total admittance of the two daughter tubes is smaller than that of the parent. So the pressure wave amplitude appears to peak, would appear to peak as the pulse approaches the bifurcation. as is observed. And that's because in the human circulation, possibly because we haven't got a tail, the total cross-sectional area of the two femoral arteries is smaller than that of the aorta. And the admittance is area divided by density and wave speed, and the wave speed doesn't change much over across the junction, hardly at all, but the cross-sectional area does. This is consistent with the observed peaking. Many other bifurcations in the arterial system appear to be well matched, so that the reflected wave is very small, by the way. Right, so the propagation of the pressure pulse is not greatly influenced by the viscosity of the blood, but blood viscosity does have a strong influence on the effect of the blood flow on the artery wall. Remember the Poissons flow picture. I just want to emphasize that the no slip at the wall means that there's a velocity gradient near the wall, and that means that viscous forces apply a frictional stress on the vessel wall. And the wall shear stress turns out to be important because it's relevant in the development of arterial disease, atherosclerosis. And the initial development of atherosclerosis is called fatty streaking. It means that there is an accumulation of lipids just inside the blood vessel wall, which therefore increases in thickness and stiffness. And as time goes on, over the years of a person's life, these fatty streaks may grow thicker and break off. Well, they first of all grow thicker and fibrosis takes place. This is a post-mortem coronary artery, the inner sections of which are not just lipid accumulation, but fibrous tissue that develops from that. And then this stuff can break off and flow downstream and block a smaller heart vessel and cause thrombosis and heart attack. But this is a picture of the excised abdominal aorta of a rabbit, I think, which had been given a high cholesterol diet, and the fatty streaks are marked by this red dye. It's not blood, it's a red dye that is taken up by the vessel wall when there's excess lipid there. And you see that it's not uniform, it's very patchy. There's a high concentration near the inside of this junction as the fluid comes along and goes in it, and a low concentration of redness on the flow divider, the far side. And it's definitely non-uniform. And it is assumed that this has something to do with the permeability of the blood vessel wall to molecules going in and out. And it has been postulated on the basis of that non-uniform distribution that the thing that makes a difference to the artery wall, the external influence that makes a difference to the artery wall, is the wall shear stress. And atheroma develops preferentially in regions of low wall shear stress. And I'm just showing this picture of the arteries again to emphasize that the geometry is very complicated, and therefore the distribution of flow and wall shear stress will be complicated. For instance, consider a nice symmetrical bifurcation when in a tube with Poissons flow coming along it, which doesn't exist, of course, in blood flow, but let's consider it anyway. And the maximum velocity, the fluid with maximum velocity is that which impinges on this flow divider. So as it goes off on either side, and remember the no slip condition, but the maximum velocity will be near this this wall of the bifurcation on the flow divider wall, and therefore there'll be a very high velocity gradient in this boundary layer, and a much lower gradient, and therefore a lower wall shear stress on the opposite side. The other thing that this shows you is that this flow is going around the bend. So, and if you flow fluid around in a curve, the higher velocity fluid will be flung out towards the outside of the bend in a sort of centrifugal force manner, and then will have to be replaced by slower moving stuff that was near the wall. And so you expect to see a secondary distribution of secondary velocities in a tube downstream of a bifurcation. And incidentally, if this curve at on the opposite wall is sharp enough, the flow will separate off, not just have a low wall shear stress, but will separate off and recirculate, and that'll slow it down some more. And this is an experiment to demonstrate the existence of those secondary motions. It's the same symmetric tube. It's actually using air as the fluid and smoke as the marker, and you look upstream from the bottom and you get these secondary vortices. That's one complicated geometry. Here's a made from a cast of the carotid bifurcation in the neck, where incidentally there's a sort of bulbous region just near the bifurcation, always called the carotid sinus. Anyway, this is steady flow photograph marked by dye, but the flow goes along until it hits the wall and then is diverted and generates secondary motions. But in this case, there's definitely a flow separation in the carotid sinus. If you've got unsteady flow in a bifurcation, if its flow is coming out, it comes out smoothly. When it goes in more or less smoothly, but when it changes direction, it goes unstable and you get an awful mess of turbulence and stuff, which emphasizes the fact that it's very hard to predict the wall shear stress, or indeed measure it. Here's another branch with a higher flow, a right angle branch with a higher flow rate off to the side than further going on downstream. And these velocity measurements show flow separation in the daughter tube and especially in the parent tube downstream of this junction. And just this is a this is going to show flow in a flow system made from imaging the descending aorta and the femoral artery main branches without other branches, but the actual geometry has led to the construction by my colleague David Koo of a set of glass tubes in which pulsatile flow, as in the arteries, is going on. This is supposed to realistic dimensionally for the abdominal aorta and the femoral arteries and the other big branches that go off. And you see that, first of all, the unsteadiness, but also the fact that flow is going off into side branches leads to considerable disturbance to the blood flow. And if you blow it up and look more closely, well, it's still very disturbed. There's very strong mixing across the vessel and instability and complication. So that's the arteries. What about the veins? I haven't got much time to talk about the veins, 10 minutes maybe. So I won't talk about them much. But remember, Harvey showing that there were valves in the veins. And in particular, I would emphasize that there are valves in the veins in the legs. And the question arises is, given that the pulsation of the pressure is highly diminished in the veins and in the microcirculation, and therefore in the veins that come through them. How is the blood pumped up? Suppose you're standing up. How is the blood pumped up from below? The answer is mostly it's given a shove by contraction of the skeletal muscles around it, your calf muscles, for example. If you're standing upright, they squeeze every now and then and push the blood up, and the valves stop it falling back. Otherwise, you get fluid accumulation in your swollen ankles. So that's the important feature of such valves. And what about, the way I've talked about it with what happens when you're standing up and standing still, and you don't want to pass out, as some guardsmen used to do. You keep twitching your muscles, and the effect of gravity is significant because the pressure in a fluid, forgetting the flow, at rest decreases with height. So over a height h, the pressure falls by a quantity which is g, the acceleration of gravity, the density of the fluid, and h, the height. And if you're very tall, this has a big effect. And some animals are very tall. A giraffe, a full-grown adult giraffe, has a height from sort of nose to feet of about 5 meters. And it needs to stick its head up to eat off these high tree leaves and whatever else it likes to do when sticking its head up, like look through hotel windows. And so what effect does this have? Is the giraffe circulation different from people? Well, it is, yes, because the arterial blood pressure at the level of the heart is about 250 millimeters of mercury. We're talking about the averages here, whereas it's 100 in people and in most other mammals. Why should that be? Well, the obvious answer is that it's got to pump the blood up to the brain, which has to have a reasonable pressure in its arteries to function properly. In millimeters of mercury, this one is the height. The formula G rho H suggests that that would have a arterial blood pressure of about 75 millimeters of mercury up there. People, it's not very dissimilar. And if you go downwards into the legs, down at the bottom, if the central blood pressure is 250 millimeters of mercury, then the pressure in the peripheral vessels will be about 400, which is twice as high as in people. If we had that sort of internal pressure, we'd get swollen ankles, enormously swollen ankles very quickly. How does the giraffe deal with that? Well, we get swollen ankles because capillaries have to be permeable to permit the transfer of nutrients and fluids between the blood and the tissues. But if the pressure gets too high, that permeability just leads to accumulation of fluid in the tissues. In the giraffe, the permeability of the capillaries is lower than in humans, and they wear what you might call anti-gravity stockings. They've got a very tight skin and fascia which cannot expand, and therefore there's no room for their ankles to get swollen. So they don't. But if you think about the venous side, well, we'll see that in a minute, but if you think about the venous side, if there wasn't any flow or anything, you'd think that because the pressure relative to atmospheric in the veins at the level of the heart is close to zero, then the pressure up towards the head would be significantly less than atmospheric. We'll consider that in a moment. First of all, let's consider the mean rate of working of the heart, which is the product of the pressure relative to atmospheric and the flow rate. So the bigger the pressure is for a given flow rate, and I'm talking about flow rate per kilogram of animal or whatever. So we assume that that flow rate per kilogram is the same in giraffes and people, but the pressure is 2.5 times higher. So the amount of work done by the heart has to be 2.5 times higher, which means that the heart muscle has to be there has to be much more heart muscle. And indeed, in adult giraffes, the mass of the heart can reach 2.3% of the body mass, whereas it's only usually about 0.5% in people. But now let's consider the microcirculation, the venous side. And this is a sketch of an idealized experiment. It's an experiment that has been done, but it's supposed to model the carotid arteries going up to the head, and it models them as rigid tubes. They're not rigid, but they're distended because the arterial pressure remains quite high, even in the head. And then it comes out of the skull and into the veins, which they're not rigid, and also, their pressure in the veins is not. If the pressure, think of the veins as a relatively thin-walled elastic tube, which is what the experiment I'm talking about did, then if it had highly subatmospheric pressure, then it would be natural for it to collapse down until the cross-sectional area is very small. If it had a much higher than atmospheric pressure in the vein, then it would remain distended, and the blood flow would be too great. Which would imply a much too greater blood flow. So if we look at the pressure difference from the pump or the heart to this exit from the skull, P1 minus P2, there's 2 contributions to it on average. One is a resistance, a viscous resistance. R is the resistance per unit length, L is the length, Q is the flow rate, plus the gravitational term rho GH. Now, in the experiment, the model experiment that was done, and the external pressure is atmospheric, and therefore the internal pressure will probably be close to atmospheric. In the experiment, it was more or less precisely atmospheric. So P1 minus P atmospheric, in order to get any flow rate at all, has to be great, positive Q, has to be greater than rho GH. So that says that to get any flow rate at all, you need to have enough pressure to pump the blood uphill. And the downflow tube is observed to collapse, and in it there'll be a balance between gravity, a gravitational force pushing the fluid downwards, and that's going to be balanced by the viscous or resistive force given by this equation here, that the RC, the mean resistance per unit length in the collapsible segment, times the flow rate is rho G. Well, for a given flow rate, that'll tell you what R sub C is, and the resistance of a collapsible tube will depend significantly on its cross-sectional area. So that'll tell you what the cross-sectional area is, which is very small. So in the case of the giraffe, especially, the model predicts that the jugular vein will collapse more or less as soon as it emerges from the skull. Or alternatively, the giraffe has a neck of phenomenal length up which blood must be pumped to the brain. The consequence is a heart of great strength and collapse of the jugular vein. Now, I was going to talk quite a bit about what happens in flow in collapsible tubes, but I don't think I will. I haven't got much time. The a standard experiment is to mount a piece of rubber tube on rigid tubes, contain the whole thing in a box whose pressure can be independently controlled, and then by raising the upstream pressure, put flow through it and see what happens as you increase the pressure in the box. And a simple theoretical model tells you, and I'm not going to go through it, but it shows that as because of the viscous resistance, the rate of change of cross-sectional area as you go downstream is negative, so it'll get narrower. But as it gets narrower, that means for conservation of mass, it means the fluid velocity will get higher. And what the theory shows, this is what's shown by this equation here, and the term, as the fluid velocity gets higher, it gets closer to this quantity c squared, which is our old friend, the speed of propagation of waves in this elastic system. And that speed gets lower as the cross-section gets smaller, because as it is collapsing, you can get quite a big area change with a small pressure change. So it's very compliant. And so eventually you'll reach a point where those two become equal and the model breaks down. And what happens is that it all becomes unstable and starts to oscillate. And the oscillations are fascinating. I'll just show you a picture of some, also taken by my friend David Koo. This is an elastic tube mounted at its ends, which we can't see, in a chamber whose pressure is gradually being increased. There's a bit of vibration, but as the pressure is increased further, you see that it starts oscillating rather dramatically. And as you go further still, the oscillations can become extremely complex. This is a fascinating dynamical system involving flow, structure, interaction, which has been examined a lot and is still being examined theoretically and experimentally in the lab. So this is an example of fascinating fluid mechanics, which is driven by experiments on collapsible tubes, which are inspired by looking at the venous flow in mammals. Giraffes do actually have unsteady flow in their veins and arteries. Well, arteries obviously, but veins. Just think of what happens, this drinking giraffe, if a leopard was coming along behind it, and it noticed, it would whip its head up 4 meters or so in a very short time, and that would require a lot of interesting control systems in the bodies for it not to pass out or for other phenomena to take place. And finally, this was a headline in a famous scientific journal, to wit, the London Evening Standard, in 1992. A group of medics had noticed that in order to pump blood up a very long neck, you need to have a very big heart. And if in a giraffe, the heart is 2.5% of body weight for a 2.5 meter neck. If you're one of these dinosaurs with a hugely long neck, it would be a 15-meter neck. The heart would have to be 15% of body weight. That's totally unlikely. And these medics said, well, what you could do if you were evolving a dinosaur was stick in a load of booster pumps on the way up, but there's no evidence that any organism has developed that sort of thing. And so the idea of these massive dinosaurs sticking their neck high in the air is out of the question. And it's much more accepted these days that they didn't, and that they had their long necks, but they must use that to sweep around the neighborhood to get food or whatever. So Jurassic Park didn't get it right, which I'm sure is incredibly disappointing. Well, I think that's it. Thank you very much for listening. Good night.
Thank you, Tim, for a really excellent lecture. I've often wondered why giraffes do not faint when they swing their heads up so quickly, if they're surprised whilst drinking, but it never crossed my mind that they might get swollen ankles. So now we all know a lot more, at least I do. We're deeply grateful to you for your clear explanation of how blood is able to flow through our veins and arteries so effectively. Next week, we move from fluid mechanics and applied mathematics to vampires and the treatment of women in Dracula with Carol Senf from the Georgia Institute of Technology. I do hope you will join us then. And thank you once again, Tim, for a fascinating talk. Good evening.